This is an introduction to metabolism. Metabolism is the totality of an organism's chemical reactions, and a metabolic pathway begins with a specific molecule and ends with a specific product, with each step in between catalyzed by a specific enzyme. For example, if you start with reactant A, a particular enzyme will allow that particular reaction to undergo and create an intermediate product, B, through that particular enzyme. And so now B can be considered a reactant for a second reaction where C would be the product through reaction number 2 and with a different enzyme. And finally, C can be considered an intermediate product and end up being the final product, D, through yet another enzyme. A term that I'd like you to know is the term catabolic. Catabolic pathways release energy, keyword, release energy, by breaking down complex molecules into simpler compounds. Cellular respiration, for example, is an example of a catabolic pathway. The breakdown of glucose in the presence of oxygen is an example of this catabolic pathway. Cellular respiration, how do you figure? Because you start with a very large molecule, C6H12O6, glucose, and we put some oxygen in it, and we break it down into smaller molecules, which would be water and carbon dioxide, and of course then you'll get some ATP out of it. Catabolic, breaking down and releasing energy. Anabolic would be the opposite. Anabolic, we've heard of anabolic steroids, to build. Anabolic pathways consume energy. You have to put energy into the system to build, keyword, build complex molecules from simpler ones. The synthesis of protein from amino acids is an example. Building. So you take something that's small, like a bunch of amino acids, and when you put them together, when you build to make a protein, this is an example of anabolic. Or you can think of the example photosynthesis. So photosynthesis really is the opposite, basically the opposite of cellular respiration, where you take smaller molecules, which would be carbon dioxide and water and sunlight, and you undergo photosynthesis reaction to make a larger molecule. You are building. Photosynthesis is building. Photosynthesis is an anabolic, undergoes anabolic pathways. And what you get for the reaction of a much larger reaction, much larger product, which would be glucose, C6H12O6. Starting with smaller molecules and coming out with a larger molecule. Building. Anabolic is building, and catabolic is breaking down. A few different types of energy. Kinetic energy is the energy through motion. Thermal energy is the energy associated with random movements of atoms or molecules. Potential energy is energy that is based on location. And chemical energy is the potential energy available for the release through a chemical reaction. And of course, any of these energies can be converted from one form to another. This example here is an example of potential energy, which is location, based on location. The person at the top of a diving board has significantly more potential energy than the people down in the lake because that person has the potential to move. Same idea, a ball at the top of a hill has the potential to, if it started rolling, could roll all the way down the hill. Whereas the ball, once at the bottom of the hill, has very, very little potential energy because it is now at the bottom of the hill. It would have to take work to bring it back up to the top, so it would have very, very little potential energy. It's not going to move once it's at the bottom of the hill. Thermodynamics is the study of energy transformations. An isolated system, such as that approximated by liquid in a thermos, is unable to exchange energy or matter with its surroundings. In an open system, energy and matter can be transferred between the system and its surroundings. Organisms are open systems. According to the first law of thermodynamics, the energy of the universe is constant. Energy can be transferred or transformed, but it cannot be created nor destroyed. The first law is also called the principle of conservation of energy. So, in this example here, you got a bear, you have fish, and through consuming that fish, the energy can be converted into chemical energy, and it now can be put to different kinds of use for building protein in the bear. And in the second, in consuming that fish, the GI tract will begin to heat up because of all the chemical reactions, and so some of that energy is lost through heat, and that would be, according to the second law of thermodynamics, every energy transfer or transformation, with every transfer, some energy is unusable and is often lost as heat. Every energy transfer or transformation increases the entropy of the universe. Here is another key word that I'd like you to know: entropy. Associate this with disorder. If you look at anything that's living in the entire world, and as soon as that organism dies, it begins to increase in disorder, in entropy. It starts to break down. It becomes more and more disordered because there isn't energy being put into the system. In order to stay organized, the organism would need to constantly consume energy from outside, whether that be the sun or glucose, for example, in humans. As soon as it dies, that organism will begin to increase in disorder, breakdown, 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 breakdown. The same idea, think of a pile of books stacked up on top of a table. Which has more entropy, the books stacked up on a table, or a bunch of books that have been kicked off the table and fell on the ground? Which of those two examples would have more disorder? And the answer is, of course, the bunch of books that have been kicked off the table and have fallen on the ground have more entropy, and you would have to put in energy in order to stack those and organize those books once again on a table. Spontaneous process. Spontaneous and non-spontaneous, I want you to be familiar with. Spontaneous processes occur without energy input. They can quickly happen or slowly. Once again, going to the example of an organism dying. Once an organism dies, there is no energy input. And so anything that happens to that body once the body has died is considered spontaneous. It just automatically, more or less automatically occurs. But in order for non-spontaneous process to occur, energy would have to be put in. How about in terms of photosynthesis? Photosynthesis is you need energy input. Energy would be the sun. You need sunlight. And if you need energy from outside of the system, that would be very much a non-spontaneous process. It doesn't just automatically occur because you need energy from outside. So photosynthesis is an example, a very good example of spontaneous process, whereas non-spontaneous, that would be more along the lines of cellular respiration. Because cellular respiration starts with a large molecule, and then it's just breaking down the molecule. Starts with glucose and then just breaking it down. For a process to occur without energy input, it must increase the entropy of the universe. Cells create ordered structures from less ordered material. Organisms also replace ordered forms of matter and energy with less ordered forms, putting in energy. This is putting energy into the system. Energy flows into an ecosystem in the form of light and exits in the form of heat. Entropy may decrease in an organism, but the universe's total entropy generally increases. We're always increasing in disorder. Biologists want to know which reactions occur spontaneously and which occur non-spontaneously, which require input of energy. A term I'd like you to know is delta G, or free energy. A living system's free energy is the energy that can do work when temperature and pressure are uniform. Free energy is the energy that can do work, and that's delta G. The change in free energy now, the change in free energy, delta is the symbol for change. The change in free energy during a process is related to the change in enthalpy, or change in the total energy, delta H. Change in entropy, delta S, and temperature in Kelvin, which is T. I want you to focus on just one of these symbols, which is delta G, the free energy, the change in free energy. Only processes with a negative delta G are spontaneous. Remember, spontaneous is negative delta G, losing free energy. Okay. Free energy is a measure of a system's instability, its tendency to change to a more stable state. Now let's look at all these things. Let's try to put these together with this, looking at this. Person standing on the top of a diving board. When this person drops from the top of the diving board down to the bottom of the lake, would this person have a positive delta G or a negative delta G? More free energy or less free energy? And the answer is a negative delta G, because the person will have less free energy as the person jumps from the top of the diving board to the bottom of the lake. Less so moving from top to bottom would be negative delta G, because they are losing that free energy. Another question you should ask yourself is, moving from the top of the diving board to the bottom of the lake, would that person, or which of those two scenarios is more stable? The person at the top of the diving board, or once they're at the bottom of the lake? Which of those two scenarios would be more stable? Answer is at the bottom of the lake. Bottom of the lake, that person is significantly more stable. At the top of the diving board, the person is much more unstable. And once again, talking about spontaneous or non-spontaneous. Jumping off the diving board, is this a spontaneous occurrence or non-spontaneous? And the answer is very much spontaneous. Negative delta G. Spontaneous. You don't have to put any energy in to just fall off the diving board and drop down into the lake. You don't have to put any energy in, therefore it's negative delta G, therefore it's spontaneous. Going from less stable to more stable. All those things, you should see the connection between them and draw connections between all those different questions. Now, how about the person having to climb up the ladder all the way up to the top of the diving board? That person would be a positive delta G, increasing in free energy. Also, that would be putting energy in, which means that would be non-spontaneous. You can also think of it as being anabolic, putting energy in, building. And also, of course, that the person is becoming less and less stable as they go up to the top of the diving board. That would be positive delta G, non-spontaneous, becoming less stable, and gaining more and more positive, more and more free energy, a positive delta G. Another way to think of delta G is to realize that it represents the difference between the free energy of the final state and the free energy of the initial state. Okay, so you got delta G on the left-hand side equals the final state. Let's look at the example of the person jumping off the diving board. The final state would have a very small G, very little energy, whereas the initial state, which is the person at the very top of the diving board, would have a much greater amount of energy, amount of free energy, because they're at the top of the diving board. Well, if you put a negative in the middle of that, a very little number minus a large number would give you negative delta G. And that's the same thing as being a spontaneous process, jumping from the top of the diving board down into the bottom of the lake. I hope that makes sense. All right, you should be able to take these 9 questions, or more like 18 questions, and ask yourself if you can answer all those questions, those 18 questions, with any particular reaction. Check out this reaction at the top. This is glucose and oxygen converting into CO2 and H2O. What is this anyway? This is cellular respiration. This is what we do in our bodies. We consume glucose, C6H12O6, and with a little bit of oxygen, we get out energy, and which is missing there, so on the right-hand side there should be energy there. We get out energy and 6 CO2s and 6 waters. This is cellular respiration. Now, ask yourself, are the reactants larger or are the products larger? Are the reactants larger? Which of those molecules is larger? Well, you add up 6 plus 6 plus 12, C6H12O6, that's a 24 atom molecule, 24 atoms. The next molecule is only 2 atoms large. You have 6 of them, but it's only 2. O2, diatomic molecule, it's only 2 atoms. The products, check out the products. A 3 atom molecule, CO2 is a 3 atom molecule. And H2O is also a 3 atom molecule. So you got 24, 2, 3, and 3. Which of those, the reactants or the products, are larger? Of course, the reactants are significantly larger, glucose. And if we break this, and if we're starting with a large molecule and we're ending with smaller molecules, we're breaking it down. And if we're breaking it down, then you ask yourself, so are the reactants larger or smaller? And in this case, the reactants are significantly larger. Question number 2, do the reactants contain more free energy or less free energy? The reactants. What do you think? Answer, more free energy. The reactants have more free energy. Are the products larger or smaller? The products are smaller. Do the products contain more free energy or less free energy? And the answer is less free energy. They're smaller. Which of the two are in a greater state of entropy? The reactants or the products? Well, there's that word again, entropy. Entropy, disorder. Where do you have more disorder? On the left-hand side or the right-hand side? The answer is right-hand side. And if you have more on the right-hand side, more disorder, then this must be spontaneous or non-spontaneous. What do you think? Question number 7, spontaneous or non-spontaneous? We're not putting in energy, we're actually getting energy out. Therefore, this is spontaneous reaction. Cellular respiration, spontaneous reaction. Going from more ordered to less ordered, spontaneous. This is catabolic, breaking down. And don't forget about 6. Is the overall reaction exergonic or endergonic? Well, do we have to put in energy to undergo a spontaneous reaction? And the answer is no. We're actually getting out energy. We're not putting in sunlight. We're actually getting out ATP, which is missing on the right-hand side there of the top reaction. C6H12O6 plus oxygen, we get out carbon dioxide, water, and actually we get ATP. So is this, question number 8, is this a negative delta G or a positive delta G? Is this negative delta G or positive delta G? Well, we're starting with a larger product, a larger reactant, and we're breaking it down. Therefore, this better be a negative delta G, less free energy, spontaneous, just like jumping off the diving board. Starting with a less stable product and ending with starting with a less stable reactant and ending with a more stable product. So, I hope that you are able to take any of these reactions. Here's 4 reactions, and ask yourself the same questions and putting all this stuff together. All we talked about was the very top one, which is cellular respiration. The next one down is photosynthesis. What do you need? You need sunlight to grow your plant, sunlight, carbon dioxide, and water. And you get out a sugar molecule and oxygen. So ask yourself the same things. But let's just go back up. Instead of answering these again, let's go back up to the cellular respiration reaction and ask these questions again. Are the products more stable? The answer is yes, more stable. Catabolic or anabolic? This is catabolic. We're breaking down. And if we're breaking down, it doesn't take any energy to break down. It only takes energy to build. Therefore, this is spontaneous. And if it's spontaneous, then this is, we're seeing more entropy on the right-hand side, more disorder on the right-hand side, because it takes energy to build and become organized. It takes energy to take books that have fallen on the ground. It takes energy to take those books and stack them up and make them organized, putting in energy, building anabolic. But this reaction is catabolic, where we have the products with more entropy, more disorder. So I hope you can put all those questions, and I hope you can take any reaction, chemical reaction, and answer all those questions and get them correct. So now we've covered all those different examples, all those different questions, those 18, and so here are the definitions of exergonic and endergonic: releasing energy, or endergonic, putting in energy. And here it is on a graph. Here it is on a graph. We see free energy, and we also see, we can also look at the putting energy in or taking energy out. Looking at example A, we have more free energy at the beginning, and the products have less free energy. We actually have a negative delta G. Would this be an example of a spontaneous reaction or non-spontaneous? Is this an example of the person jumping off the diving board, or would this be an example of a person climbing up the ladder? What's the answer? Answer is jumping off the diving board, having less free energy at the very, very end. But they get out energy. This is exergonic, getting out energy. Really? Getting out energy? Well, yeah, because the person had started with potential energy, and through the actual diving process, the person converted that potential energy into kinetic energy, which is the motion, having the energy in through motion. So I want you to be able to take all of these reactions and ask yourself, which of those four are these? Is it the example of A or example of B? Putting in energy or taking energy out? Exergonic or endergonic? Cells are not in equilibrium. They're open systems experiencing a constant flow of material. We'll see later on in later chapters how we'll see this example of energy coupling, which is the use of exergonic process to drive an endergonic one. Most energy coupling in cells is mediated by ATP. This molecule you need to get familiar with really, really well. ATP, adenosine triphosphate, is the cell's energy shuttle. This is the dollar bill of the human body. ATP, this is the dollar bill. It's actually not glucose, it's ATP. This is the energy currency of the body. You have three phosphates, a ribose, a sugar, and a nitrogen-containing base. ATP. Well, this is the high state of the energy, but it also can be broken down into ADP, adenosine diphosphate, and even further down into adenosine monophosphate. But we generally look at this thing as ATP breaking down into ADP and then back and then vice versa. So if we go from it doesn't try ATP to ADP. This is where we are releasing the energy in the bonds of the ATP, and so we get out energy. Exergonic. We're not putting in energy, we're getting out energy. And just as we saw before, this would be hydrolysis, also an example of hydrolysis, adding water to break apart a molecule. The bonds between the phosphate groups of the ATP tail can be broken by hydrolysis. Energy is released from ATP when the terminal phosphate is broken. This release of energy comes from the chemical change to a state of lower free energy, not from the phosphate bonds themselves. If you want to get technical, the energy technically isn't in the bonds. It's actually from the chemical change to a state of lower free energy. But usually when we talk about it, we just make it easier on everyone and just say it's in the bonds. So if you see a molecule with 15 bonds, 15 atoms large or 16 atoms large, it has more energy than a molecule with, for example, 3, because the energy, but to be very, very technical, it's actually not, the energy is not in the bonds themselves. It's just from changing from the chemical change to a lower state of free energy. But we don't need to get hung up on that. The 3 types of cellular work, mechanical, transport, and chemical, are powered by the hydrolysis of ATP. ATP drives endergonic reactions by phosphorylation, transferring of a phosphate group to some other molecule like a reactant. You look at this diagram here, and if we are to take at the bottom there, for example, you got a smaller molecules, ADP and phosphate, and if we are to build it into ATP, coming out with a larger product, we have to put in energy. And if we put in energy, this is called an anabolic process we're building, and so this would be considered endergonic, putting in energy. And if we start with ATP and break it down into ADP, we get energy out, and that would be considered exergonic. A catalyst is a chemical agent that speeds up any chemical reaction without being consumed itself, and an enzyme is an example of a catalyst. Enzymes are the catalyst of the human body. Hydrolysis of sucrose, for example. Hydrolysis of sucrose by the enzyme sucrase is an example of an enzyme catalyzed reaction. You got sucrose, which is a two compound molecule. This is a polymer. This is a disaccharide. And if we're to break it down, we need an enzyme. This is the first time now that we're gonna see enzymes. We always talked about before adding water and undergoing hydrolysis, but actually it also takes an enzyme to break it down. And so anytime you see a word with the ending ASE, that is the name of the enzyme that breaks down the molecule itself. Sucrose, sucrase, for example. So there you have it. You have a disaccharide undergoing hydrolysis through the enzyme sucrase that doesn't get consumed itself, but it speeds up the reaction and allows the reaction to move forward breaking up into glucose and fructose. Well, the activation energy is the initial energy needed to start a chemical reaction. You see that hump? The hump between the reactants and the products, how it goes up, that's the activation energy. Well, enzymes lower the activation energy, allowing the progress of reactions to move forward, allowing the reactions to move forward. Enzymes lower the activation energy, allowing things to move forward faster. Without enzymes, reactions will still move forward. It would just take a very, very, very long time. So we need these enzymes to lower the activation energy, lower that hump. Imagine a ball, you're starting at the top of a hill there, but then you got another little mountain. You roll the ball, and you're on that black hill. It'll take a lot of force to roll. If you're bowling, to roll up that, for a ball to roll up that hill, and then of course on the way down it can do it easily. Well, if you cut out a whole bunch of that mountain and you try it again, it won't take as much energy to bowl and roll that hill up, and that's what enzymes do. They lower that mountain, lower the activation energy. The active site on an enzyme is the region where the substrate binds, and the substrate is the reactant that an enzyme acts on is called the enzyme's substrate. So you have a substrate, you have an enzyme, and you have an active site. If the substrate fits in the active site of the enzyme, then you have a match, and the enzyme can convert that substrate into a different product. In an enzymatic reaction, the substrate binds to the active site of the enzyme. The active site can be lowered by an activation energy barrier by orienting substrates correctly, or straining substrate bonds, or providing a favorable microenvironment, or covalently bonding to the substrate. And here's an example of that. pH and temperature also are also huge factors in how well enzymes will work. And so, for example, a typical human enzyme's optimal temperature is likely to be the same temperature of the internal body of the human, 37 degrees Celsius, 98.6. It makes sense. Otherwise, it wouldn't be there. And the same idea, an enzyme in the stomach that's supposed to be in the stomach to break down foods has an optimum pH of 2, which makes sense because that is the environment of the stomach acid. So it makes sense. So if that enzyme were to move out of the stomach and into a different area where you'd have a different pH, the enzyme would then slow down in its work because it's now in an environment other than optimized for what it likes. So pH has a factor, and also temperature has a factor in the rate of reactions that an enzyme can undergo. There are different types of enzyme inhibitors. There's competitive inhibitors that bind to the active site of an enzyme, competing with the substrate. There's non-competitive inhibitors that bind to another part of an enzyme, causing the enzyme to change shape and making the active site less effective. So if the normal binding, you have some kind of shape of the active site of the enzyme there, and you have so then the substrate can bind normally, but if you in competitive inhibition, you can have another molecule that is able to fit and inhibit the other substrate that could normally bind. This is called competitive inhibition, where you have an inhibitor that can move in the place and fit into the active site of an enzyme, slowing down a particular reaction. And in non-competitive inhibition, the inhibitor will do the same thing, except instead of moving into the same active site, it can actually fit into a different active site somewhere else on the enzyme, and yet change the shape of the active site, of the regular active site, and cause the regular substrate not to fit properly, and therefore inhibit the reaction. So yeah, that's the difference between competitive inhibition and non-competitive inhibition. In allosteric regulation, this may either inhibit or stimulate an enzyme's activity. Allosteric regulation occurs when a regulatory molecule binds to a protein at one site and affects the protein's function at another site. And so here we see how we have activators and inhibitors. So on the left-hand side there, the top left-hand side, we have an allosteric enzyme with 4 subunits. We have an activator, and now we go from unstable to stable. And the enzyme itself can also fit an inhibitor. It can fit an activator or an inhibitor at a different place, and that's why it's called allosteric. It can undergo both inhibition or activation. In feedback inhibition, the end product of a metabolic pathway shuts down the pathway in the initial pathway. So if you move across and down the right-hand side, where you have enzymes working, enzyme 1, 2, 3, 4, 5, you'll make these end products, for example, isoleucine. And when you make and the products themselves inhibit the actual pathway. And that is an example of feedback inhibition. We're moving forward making these products, but the product itself will also be able to regulate the pace at which the enzyme makes more and more isoleucine, for example, because isoleucine can bind to the activation site, the allosteric site, and slow down the rate of synthesis. The initial reactant is theriane, for example, and then it's converted and converted and converted, and the end product is isoleucine. But if we have isoleucine in the way inside the active site, then we'll slow down the actual reaction that it initially started. This is called feedback inhibition. One way that cells can regulate the rate at which they make products. And that concludes the introduction to metabolism.